Okay. Hi, I'm John Quinn. Let me point the right way again. Director of Science and Product Development at BD Biosciences Informatics, aka Flojo. My contact info is there, john.flojo@bd.com. Feel free to write me with questions and comments. And with that, welcome. Let's go. Let's do this. I am going to share my screen. Okay. And there we go. And let's go to presentation mode. Okay, so easy Dafi, supervised clustering for Flojo. And now actually advancing the slides. Okay, so the big picture here is, I think, something that most people will be pretty familiar with. You've got some data, and it varies a little bit from sample to sample. And this could be because there are slight differences in people. This could be there are differences in cytometers or across time points, or a number of reasons. And so you've got some kind of gating situation that looks like this, where clearly the setup on the right, those cells look like they're all part of one population. And so we'd probably like to adjust that gate to include the cells within the contour lines. Just a little bit variable from the first. You could make the first gate like wider and more encompassing, but wider and more encompassing isn't always going to work either. You may just get more junk. So we'd like to have some kind of tool that will auto adjust the gate on the right to be appropriate for that data. Some of the tools that folks might be familiar with that we've prevented at various times in the past, there's been a magnetic gate tool in Flojo for forever. And that works pretty good. It really does in any situation where you've got a cell population that's pretty consistent in shape and even density, really. So that's just an example where if you have the population changes shape. Here we see in just an example from a data set where a population gets wider. There's a little more spread in this one. You'd like that gate to adjust and get wider to match the data. So there are places where magnetic gates are good, but they don't handle size changes. And then there's unsupervised clustering, which will accommodate every sample uniquely and just give you a pretty nice division for the data without any intervention or heuristics. But sometimes we want to intervene. We want to look at these two samples shown in the clustering example and just know that the orange population here is not the orange population there. That we've hit a sample where we're just missing those populations. The orange and the light blue should probably just go away at that point. And we should not be trying to compare whatever we can find to them. And so a little bit of supervision mixed with a little bit more flexibility is where Easy Dafi fits in. Easy Dafi stands for, well, the easy is just for easy, but directed automated filtering and identification of cell populations. Okay, so what that means is that it is going to allow you to direct what the filter should be for which cells should be included and then do clustering within a data set. So it's recursive filtering. So there's multiple steps. It'll do clustering and it'll be able to be constrained via manual gates. So this is based on a paper. Let's see, I think I first saw this presented probably at Cyto up in Vancouver, where Max, the last author on that paper, came up and did a nice talk on it. And he came and hung, sorry, making my tenses right. He came and hung out with us for a while, Flojo, and then ended up building a plugin that we could use based on this work with a little help from our plugin development team. And so it's available on the Flojo exchange right now. The gist of the algorithm, as shown directly in the paper, I've taken this, this picture directly from the paper. So you've got some data. And then we can use some kind of clustering method. And the default is FlowSOM, because FlowSOM's awesome and we love it as a fast, pretty effective tool for many, many things, to do some just clustering and say, okay, so it looks like just based on the shape of the data. So FlowSOM, the SOM stands for self-organizing map. So we let the data self-organize and we say this is where it would fit into a map. Looks like there's these populations, 6 or 7 populations here, annotated by color. And then we take those populations and look at where your manual gate is. And so this is a fancy picture shown in 3D, but really we were going to be totally fine with taking a rectangular gate, an ellipse gate, whatever you draw in Flojo on a 2D space to say, these are my T cells, B cells, whatever it is you're looking at. So it's just abstracting the idea that you've got a gate that says, this is where I think the population is. And then Easy Dafi will say, so, okay, we'll include all the cells for the next step where the cluster centroid for that population is included in that gate. So it could be that, the cluster centroid for the blue cells, for example, not every one of the blue cells is in your initial manual gate, but because the centroid is, we're going to consider all of the blue cells as potential members of your actual population. And then so we will do another clustering using multiple dimensions. So not just the two that you've drawn your manual gate on, but multiple other dimensions. And one of the really cool things about Easy Dafi is that it will look at the rest of the dimensions that are available. And you don't have to tell it which ones to include. It'll figure out which ones have the most discriminatory power and automatically include them. So you just give it a number. How many extra dimensions should I include? And it'll pick the most impactful ones and use those. And then it produces an adjusted cluster here where, perhaps a clustering algorithm would have divided these up, but you as the user saying, for my purposes, the scientific test that I'm trying to run, those are all one type of cell. And where are the edges of that population? And you let the clustering algorithm basically set your edges. So that's the plan. There is some setup involved in running Easy Dafi. It is an R-dependent package. Max wrote, or Max and his team, sorry, I know Max, but there's many authors involved, wrote it as an R tool. And so you would download this off the exchange. You would need R on your computer. And then you would copy and paste this line of code into R. And then additionally, if you're on a Mac, it requires Xcode and Xcorts, which are done by going into the terminal and pasting in this line. Or if you're on a PC, the equivalent is RTools, where you download that from the CRAN project and install it. This is all in the how-to PDF that comes with Easy Dafi. When you download it, it comes as a zip file off the exchange. You'll get the program. You'll get the how-to instructions. It's essentially a download and a couple copy pastes. So sorry if this looks frightening and scary because there's like a little bit of code involved here, but it's not too bad. And then as I called it here, there's an excellent help video. I believe that is also linked in the help documentation where my colleague Dylan put together a how-to on getting this thing up and installed. And so you can watch that too. And that's all available in the download. So you don't have to frantically write this or screenshot or whatever. You can probably just open up the download and directly copy paste out of the PDF to get the installation instructions. Just want to give you a heads up that there's essentially one extra step for using this tool compared to a lot of the R-based tools. Okay, when you run it, you will end up picking a population that you're interested in having Dafi just everything downstream of, and then it'll bring up this menu. And so the top line is the important one, where it'll say, how many additional clustering dimensions should I use? 3 is the default, seems pretty reasonable. It'll ask you which algorithm to use, flow sum by default. You could uncheck it to use k-means, but I think you really want to leave that checked on. I think flow sum is a much nicer tool. We generally recommend folks leave the R script on so that it can show it upon completion, keep it as a text format. If anything goes wrong, you can just send that to our tech support and we will generally be able to help you out with what went wrong. We've got a few extra experimental options. You can leave them off. I don't know how much they help really, and it slows it down, and the program works pretty well without it being turned on, so why? And then down here, we have just some choices for how would you like your output plots to be represented. And so I tend to turn both of those things on, up to you. And then there's the paper. So if you want to get the actual link, it'll be here. It'll also be up on the exchange. Okay, one other key factor, Easy Dafi modifies and adjusts it creates a new gate, an Easy Dafi version of each of the gates that are downstream of the population you pick it on. But it gets unhappy if you try to overwrite an existing Easy Dafi gate. So the way that we're going to do this is we're going to take one exemplar file and say run Easy Dafi on it, and then drag it to another group that the exemplar is not part of, so that it does all the adjustment for all of the rest of the files. It'll throw an error at you if it tries to overwrite an existing Easy Dafi gate. I think it creates some kind of infinite loop. It's the inception of running a FlowJo plugin, where this is an algorithm within an algorithm. I don't know. So anyway, I'll show you once we open up FlowJo exactly how to do that. Okay, so an extra step for the install. Running is straightforward. You can open up a menu and generally hit OK, and it'll do its thing. So with that, demo time. And I did promise to look at the chat. Let me make sure. Cool. So far, all the theory, all the background must be crystal clear. No questions yet in the chat. And we'll close this up. Okay. And just bring up a FlowJo workspace. Okay. So the gist here again is I've just got a bunch of files. I've put some pretty standard cleanup gates on them. Feel fairly confident about that. But perhaps some of my just top level phenotype gates, B cells, T cells, NKs, and maybe some of the activation markers on them, maybe they vary just a little bit from sample to sample. And I don't know if my standard just big rectangular gate for CD27 positive will be great for every sample. Maybe I would like to move that one up a tiny bit. And on this sample, there aren't any, must be a control here. And so, maybe clustering wouldn't be a great choice. Here's one, again, maybe up. This one, maybe you want to be down a little bit, maybe this one a little bigger and so forth. So there's a bunch of these files that you probably like that gate to vary just a little bit. And then we've got a whole slew of gates that we'd like to do the same kind of thing with, where it'd be ideal if somebody could look through and adjust each of those gates just a little bit to make sure you have much more natural looking boundaries, that you're not cutting off what seems like a continuous distribution, just because that sample varied a tiny bit. And that's where Easy Dafi is going to come in. So as I mentioned, I have this set up where I have, of my 10 files, 9 of them are just in this group together, and one of them I put in a group just called exemplar, where I've got each of the gates right here. And if I say, you know what, time gate's fine, maybe I ran flow AI on it to produce a good event, bad event gate or something like that, cool. Live gate's pretty straightforward. But right here, when we get to the white blood cells, everything downstream of that, I would like Easy Dafi to work on. I'll just click on that population. And so it's going to work on these 1, 2, 3, 4, 5 populations, workspace, plugins, Easy Dafi, sorts to the end of the list alphabetically because the easy is lowercase. Okay, same menu that I showed you in the slides pops up. I'm going to just stick with the 3 additional clustering dimensions. One of the nice parts about the algorithm is it'll actually show you in the output which of the dimensions made the most impact and how big an impact it made. Sticking with FlowSum. Incidentally, you can mouse over any of these. If you don't remember what a particular choice was, mouse over it. There's text, there's tooltips everywhere as my computer is showing for us. And I'm going to turn on the overlays. Why not? Let's get the extra picture and say, okay. Okay, so this is going to think for a little bit. It's going to take about 50 seconds to go through and create Easy Dafi gates on each of these 6 populations. We'll know it's about done when it starts popping up graph windows, making gates and producing a layout for all of them. So in the next 30 seconds or so, I will check the chat, see if there are any questions, and get a sip of tea here. All right, chat is good. And we won't have too much longer to wait. And I guess I'll take this time to mention, I do feel like the applications are when you've got a specific panel to run and you want to know the outcome of particular types of cells. And you do feel like there's a little bit of variability that you want to account for. So that's when I would most often use this particular tool. Okay, so there we go. All right, so you'll see what the output is there. Just a set, there we go, we're starting to make our pictures too. But importantly, a set of gates with the same name as your gates, just with Easy Dafi preceding it. So you know which ones are the Easy Dafi, which ones are the original gates. Upstream, you'll get a series of derived parameters. That's what that A/B is. It means it's a derived parameter. So essentially it ran the clustering algorithm, and said, once I've now done the recursive bit where I've factored in the gates and the clustering and so forth, which cells should be in the B cell population, and it made a derived parameter that just says in and out, and gates on that derived parameter for in versus out. Okay, and then it pops up a series of plots like this, where it'll say something like, okay, here's our comparison. So now you get a gate where you see that there's some overlap here, where we've clustered and found that these cells here that would have been excluded previously on the original, I should have just opened up the original gate. I mean, you can probably see that too. So sample E1, and that is our WPC. Let's go pseudo color. And maybe that was like the 3 verse 20 population. Okay, so that would just have like a hard edge if we just drew that gate typically. And it looked at these cells and said, you know what, looking at all the different dimensions that I am interested in, and particularly CD16, NKG2, IgG, those were the ones that were most important. They've got the highest T-score for impacting whether they were in or out. Those look a lot more like the T cells here than they do like the double negatives. And so it included them. And it's done that for each of these, and you can just treat these as flojo populations that you could make your own overlays if you want. It's got all the downstream ones there as well. Okay. And then if we were going to apply this to the rest of the data, I would just grab easy Dafi of white blood cells and drag and drop to white blood cells for the group and rerun it. And then it would apply it to all of the files in that group. So it's roughly 50 seconds a file, so you don't want to wait 9 minutes. And so I'm just going to open up easy Dafi finished. Oh, and there is a QA. What means 3 clusters in the plugins option. I think that's 3 parameters, I believe. And that was the additional parameters to consider when you're doing the clustering. And so again, it's not easy Dafi is going to look at the data, the gates and all that, but then consider additional dimensions for where should I put the, effectively, where should the boundary be? And that's what that, so if we go back over here, that's what that T-score is showing down here. It was saying which parameters had the most impact in terms of deciding whether these cells should be in the gate or out. And so clearly the most important ones are going to be the ones that the gate's drawn on. But then CD16 apparently was highly differentiated here between ones in and out. And so we ended up drawing a tail that went a little further in and including some of the dots in here and some of the dots there and not the rest of it. Okay. All right. Let's start. Where did finished go? There's finished. Okay. So now same thing. It just has for this particular file, easy Dafi of white blood cells, and then the easy Dafi version of all the gates. And so it just runs, you just watch them pop up one after the next as you go through the samples. If we go over to the layouts, we will then have a bunch of layout pages, 1 per. If you've checked on the box for yes, please make a layout for each one of the files. If you run this a few times and you get bored with the novelty of having it do like a report page for each of your samples, uncheck the box and then you won't end up with a ton of layouts. For 10 samples in this demo experiment, that was totally fine. For, I don't know, 100 different events, maybe a little, 100 different files, maybe a little less fine. You just want to make one summary plot. So I've done that here. And here's just an example of CD27 gates. And so you see if I'd use the rectangular or ellipse gates, you end up with populations that don't look quite so natural. And then here on the right are the easy Dafi gates where it's actually, again, used five dimensions to figure out which cells look most similar to your exemplar inside the gate and have drawn what looks like a much more natural division of the cells. Sorry, let me turn that off. Okay. And as trivial as this particular file looks, I keep pointing out that there's a really nice advantage to a semi-supervised clustering algorithm compared to a actual just completely unsupervised clustering algorithm, in that it would have had to say that these cells here are something. There's a population there, even though there isn't. That sample just doesn't have any. So anyway, here is easy Dafi applied to all the rest of the files. I think I had one other top level summary batch that just showed all the just higher level phenotype gates laid out in order also. Boom, 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 boom. Okay, so there you go, just in 3 colors. Okay, so again, just showing overlaying my manual gates, you end up with fairly artificial looking boundaries. Using easy Dafi to suss out the borders of all the populations, get much more natural looking boundaries in the data. Okay, so that is really all there is to it. I'm going to hit answered live on that question. I think it's a nice tool to address a specific niche or niche, and I'm happy to take any more questions on that. Can I set up a template with this that also has a layout with batch layouts analysis that will then apply Dafi to samples dropped into the template? So Thomas, yes, you will have to do the drag, and it'll work again as long as the populations have the same name, is basically what it comes down to. So templatized, that should work then. So it's the same kind of principle as using Flow AI in a template, in that we are creating these derived parameters that are, oops, it's the parameter itself. I should have gone anywhere in here where there's a population and just pointed out that we've got like these derived parameters, show it as a histogram that we're just doing like a gate on to say like in or out of a particular cluster. And so as long as you've got the same names, you're just getting this gate that says, yep, we've clustered it, we've figured out that should belong to that population it's in. You will still have to do on the new samples to drag the Dafi node to the group to run the algorithm on everything, but it'll then fill in events that match, like the gates are trivial. The gates are just, as I just showed you, they're just a range gate on a histogram. So with the names the same, it'll just fill in the events that are part of that gate. And then if you've made a pre-configured layout that has the samples in the order and the orientation you want, it'll just fill that in. So that's a good workflow. There's another question. It applies what's calculated on the exemplar to draw gates on the rest of the group, or will it recalculate this for each sample thereafter? Every time you run Dafi on a sample, it's taking the exemplar on that sample and using it to do the adjustment. So it is a per sample tool at that point. So that's why it's going to work a little bit better than something like a magnetic gate that is just going to take the exemplar and move it around a little bit, but that's that. And let's see. So what about, Juan asks, what about tables? I don't see an issue with tables. If you've got a population in Flojo, whether it was derived by EasyDaffy or otherwise, you can drag it and drop it into the table, create your table, and go from there. So a couple of blanks, but we know that there were a handful of controls that didn't have a particular population. So that's what I will assume those blanks are. And so that would apply for I'm assuming you're getting at like the template approach too. So again, assuming you've got your populations named the same, which makes the EasyDaffy parameters come out with the same name, then all of this should be templatizable. Okay, we have got a question from Kevin that says, could one supply hyperfinder derived gates as the exemplar gates? Yes. There's nothing special about the gates that I put on there. I drew some rectangles, I drew some ellipses. Hyperfinder, depending what your selection is going to make something like a polygon. That's fine. What all we're really doing is, putting in a boundary and saying, make sure that the centroid is within that boundary. And so if you've got a hyperfinder gate, that can be used as the boundary. So yeah, sure, no problem there. Let's see. Anonymous, ooh, mysterious, asks, if I want to use EasyDaffy as a one-time thing to help decide best way to draw a gate, how do you recommend implementing EasyDaffy generated gate back to a traditional Flojo analysis? Okay, well, I think the answer to that question might have been in the previous question, whereas any clustering algorithm is going to, it's not going to have a geometric gate as the output. It's just going to have a cluster membership. Is this cell in cluster 1 or 2 or 3? Hyperfinder is the tool that we typically think of as taking, some kind of non-geometric gate and putting it into a geometric form for sorting, but you could use that for anything you want. And so Hyperfinder will give you a geometric equivalent of the cells that were in it. It's going to draw a series of gates. And so maybe, just to be clear, you wouldn't get one gate back that recapitulates exactly what EasyDaffy did. You'd probably get a series of them because it's going to try to work in multiple dimensions. And that's how it'll manage to come up with those perhaps overlapping distributions. But I think, yeah, those two questions dovetail very nicely together. Hyperfinder's got some ties into this. Juan says, would it be better with concatenated samples? Depends on your experiment, Juan. So I think one of the real benefits of Easy Dafi is that when you drag it to a group, it'll work on each sample individually, making minor adjustments for that sample. So you don't have to concatenate. However, if you've got some kind of experiment where perhaps there's another level to this thing, you've got some meta level to the data where you've got samples from multiple patients, but also multiple time points, and you want to set up an exemplar on one time point and apply it across all the different time points, then perhaps concatenating all the patients together, that probably expedites the process a little bit. I don't think the number of events in the file impacts the time to run nearly as much as the number of files. And so you might have a little quicker run. You might have more dense data to work with. It's just generally nicer for a clustering algorithm to have a better sample size and so forth. So perhaps is the best answer I can give you to that one, depending on your experimental setup. And if you're really just looking to make individual assessments on a whole bunch of different files, yeah, I guess you could still maybe have it run faster by running it on just one, but then you would lose some of the individuality about it, but you could then break them back apart afterwards by deconcatenating. So maybe. Let's see, Maria says, is it possible to apply Easy Dafi after running downsample? Yeah, you could do that. Again, so Easy Dafi is just going to take whatever population you fed, and I didn't do it at the top level. I picked just some gate in the middle of my gating stream and applied it there. And so if, instead of making like a live or a whole or a time gate upstream, you had a downsample gate, sure. It's just one more way to filter out cells that you're not interested in for whatever reason, just the too many perhaps aspect if it's downsampled, and show Easy Dafi some exemplar and say, figure out what the boundary should be on this example. These are great questions, by the way. Keep them coming. I'm happy to chit chat more. Questions mean maybe we showed you something that inspired your imagination a little bit, got you interested in using the tools. I'm happy to see them come in. And it was a short webinar so far. It's like, hopefully because it's very straightforward and easy to use, and we didn't have to get very deep into and then click this, drag this, apply that. But it was really just go and run. And so we have the time if anybody has further questions. Let me go back. Everybody put them in Q&A. You guys were really Zoom savvy, I guess, as everybody is at this point, as opposed to the chat, which is nice. Okay. We look like we have got all of the questions answered. If you've got more, type them in. Oh, we have a raised hand. Okay. There we go. Let's see. Thomas says, I assume more parameters will just add more time to the calculation. Oh, chat was disabled. Weird. All right. Well, seems like the Q&A is working just fine. we'll stick with that. And we'll just turn off the chat then. Why bother? Okay. So Thomas, I don't, I can't say that I've really benchmarked it. I do know that Easy Dafi is calculating the importance of all the parameters right off the bat, essentially like principal component analysis. It's saying which ones have the most impact based on which ones contribute the most to the variance. So that's already calculated. It's just a matter of them clustering on. I don't know, if you cluster on a few more parameters, I guess it'll take a little bit longer. I would say, if I had to guess, yes, it will take a little longer, but I would guess it's not particularly impactful unless you're going from like 3 to 40 or something, some massive increase. I do like the fact that you don't have to worry about which ones that it's going to feed the algorithm, the ones that are most important. I would say you have to use at least 3, or else I think the 2 that are defining your manual gate are always going to be included. So you're really just working on them if you don't. But yeah, at least 3. And depending on how many parameters total you have, you may want to include more. I think it's also because you get a score for each one. I think it would also be interesting. Let me go back up to one of these that actually had the Easy Dafi summary. I think it's worth looking at maybe your first run at it and checking out the score and seeing if there's a big drop somewhere. Like perhaps we wanted to have said, give me an extra one, because I guess I took 4 when I actually did when I did the 4 run here. That 232 and the 266, there's still relatively big. The big drop off comes when you get to about CD20 there. And there's definitely, a point of diminishing return with the extra parameters that you add to it. All right, I'm going to click answered and answered. Other questions? All right, we may have handled them. I will give you back 30 minutes of your life if that's so. And if another question comes up in 5 minutes after we hang up, let me point back over to where I'm got my picture still going, which I kind of forgot about randomly here. All right. And just point out that my email address is just right there, just john.flojo@bd.com. And I'm happy to continue the discussion on this one. Ooh, we've got one more. Is Flojo compatible with all different models of flow cytometry data files? So yes, Flojo will handle any FCS file, flow cytometry standard file. At this point, I think I can say every or almost every well-known cytometer manufacturer puts out FCS compliant files, and we read those. Your acquisition software might give you the choice to export an FCS file or some kind of proprietary file. You could do like Miltenni's MQD file, something like that, which we handle, but we handle FCS files better. That's just the standard file that everything in Flojo is designed to handle. So as long as you can export an FCS, we can do it. We can also just take in a CSV file. If you just drag in CSV, we'll convert it to FCS. So in most cases, that works fine. So SpectroFlow, I believe, is Cytek's acquisition software. So yeah, we handle Cytek data too. You can either choose to unmix on the cytometer. You could unmix in Flojo. Either way is going to be fine. I think with the Aurora, it's a little nicer to do the parameter labeling right on their software. So maybe you want to do that there. But at the point where the data is unmixed or compensated, or take your whatever system you're working on, whatever kind of compensation or unmixing you're doing, once you've done that step, it's effectively just a spreadsheet, no matter what, a list of cells versus a list of parameters, and we handle that. With the .fcs, pretty much just telling you, what's going to be in the header, so that we can find things like, oh, what'll be the range and the bitness of these parameters, so we can represent them well. So yeah, whatever cytometer you want to use, that's fine. All right. Other questions? Roll. All right. Why don't we wrap up for today? Thank you all so much for joining. I appreciate the good interactions. Was even if done via typing, it was a good conversation about this tool. I hope you give it a try. Let me know if you do. Let me know if you have questions with it. And with that, I'm going to stop sharing.